The overall goal of the following experiment is to obtain protein complexes from living Drosophila embryos using an in vivo cross-linking approach. This is achieved by first loading thousands of flies into a population cage and collecting early embryos. Next, the proteins are cross-linked in vivo with formaldehyde, which stabilizes their internal protein complexes. Then, protein extracts are prepared from the cross-linked embryos so that the protein complexes can be immunoprecipitated. The results identify the isolated protein complexes by Western blot analysis.
The main advantage of this technique over existing methods, like traditional isolation of protein complexes with in vitro approaches, is that this in vivo approach allows us to capture transient but functionally significant protein-protein interactions which might not be detected with traditional in vitro methods. Demonstrating the technique will be Dr. Ming, a postdoc from my laboratory.
First, mix water and fly agar with a stir bar. Autoclave this mix. During the autoclave run, mix sucrose into apple juice and heat this to 70 degrees Celsius on a plate. Later, combine the mixtures and allow them to cool for 15 minutes. Before the agar solidifies, make fresh methyl 4-hydroxybenzoate in ethanol. This is the preservative. Add it to the solidifying mixture and stir it in for 5 minutes. Then pour the mixture into 300 square centimeter plastic or styrofoam plates without forming bubbles. Store the plates at